Hi guys! Do you know we can use molecular techniques such as cytogenetics to help in conservation of biodiversity? How, you may ask? Let's let them explain.
First, let us understand what cytogenetics is. Cytogenetics is the study of chromosome that give researchers access to high-quality chromosome preparations. Here is an example of metaphase chromosome plates and karyotypes of a species of rainbow shark. This karyotype can be used to detect anomalies in chromosomes. This greatly improves our understanding of the genetic makeup, evolutionary history, and also the systematics of the species. Hence, it is used to suggest ways in which the loss of genetic variation may be minimized.
So let us move on to the cytogenetic analysis and its application in conservation genetics. Cytogenetic analysis can be applied proactively in translocation program by screening the translocation stock for karyotypic variation. So what is translocation program? It is defined here as the release of animal to establish, re-establish, and also for augment existing population. And what is karyotypic variation? Let us look at the next slide.
So in this slide, it shows the karyotypic variation between these animals.
Retroactively, they can prove useful in the case of hybridization at both interspecifics and in chromosomally polytypic species, in intraspecific levels as well as in population, showing well-documented evidence of reduced infertility and also perinatal mortality. Cytogenetic analysis has become an essential part in the management of captive mammals. Without such planned investigation, one can confidently predict that unnecessary hybridization and reduced fertility will frequently take place in the captive group. It is similarly important that those animals are to be released again into nature. Only cytogenetically similar animals we use.
The basic elements of fish are DNA probe and a target sequence. So before hybridization, the DNA probe will be labeled by various means such as nick translation, random prime labeling, and also PCR. So two labeling strategies are commonly used, which are indirect labeling and also direct labeling. So for indirect labeling, probes are labeled with modified nucleotides that contain a hapten, whereas direct labeling uses nucleotides that have been directly modified to contain a fluorophore. So the labeled probe and the target DNA are denatured, and then combining the denatured probe and target will allow the annealing of complementary DNA sequences. So if the probe has been labeled indirectly, an extra step is required for visualization of the non-fluorescent hapten that uses an enzymatic or immunological detection system.
Next, we move on to the case study. Rock wallabies or genus Petrogil are iconic Australian marsupials that form part of the kangaroo and king family. There are currently 70 described species and 23 chromosomal releases or subspecies, including 5 endangered and 7 vulnerable taxa, under the Environment Protection Biodiversity Conservation Act 1999. Of these, 6 species have had or have ongoing conservation management, including captive breeding programs, translocation, and reintroduction. This figure shows the illustration of chromosomally diverse Australian East Coast rock wallabies, or we call it Petrogil. A shows the distribution map of the species. B karyotypes of 8 of the East Coast species, highlighting arrangements including fusions 6 to 10, 5 to 10, and 6 to 9 inversions, and variable karyotype morphology of chromosome 2 and the X chromosome. The nucleolus organizer region highlighted on the X chromosomes by white lines. C. Mapping of chromosome 3 genes, e.g. FLA in red and TMPRSS2 in green. The location of these genes demonstrates that a rearrangement of chromosome 3 has occurred between these two species, highlighting the value of cytogenetics in determining gene order.
Hi, my name is Farhanah. I will proceed to the limitations of cytogenetic. One of the limitations of this technique is the shortage in trained cytogenetics, as some of these methods require to be combined with other methods that are more technological advanced that need a good expertise in handling the technique. Besides that, this method is difficult to handle in large sample of size. This is due to some method in this technique that does not suitable to be used in large sample of size. Thus, it will cause the usage of long-term tissue culture that is more expensive and difficult to be maintained.
As a conclusion, there are many hypotheses of cytogenetics. As we know, cytogenetics originally used to determine differences in chromosome number and morphology between species. Cytogenetics can identify chromosomal differences not only between closely related species, but also between populations of different species and how these differences could impact management forecasts. Cytogenetics plays a key role in the detection of chromosomal abnormalities associated with malignancy, as well as the characterization of human alteration that allow more research and increased knowledge about the genetic aspect of the disease. Molecular cytogenetics approach such as fluorescent individualization, where either a small segment of the genome or an entire chromosome are functionally labeled and hybridized to chromosomes, have given more precise information on the extent of chromosome arrangement between species. Lastly, many benefits can be gained from combining cytogenetics and genomics. That's all from us. Thank you.